This is a very interesting collection. This very interesting collection that my research scholar Rosie D'Souza has brought to the mycological laboratory in Goa University, Department of Botany, on July 20, 2015. We are looking at various stages of Termitomyces, and there are at least 2 to 3 different species, beginning with this one, which is closer to Medius or Gomandakiensis, some of the smallest, to what we call as irises, Termitomyces, to we can see this is Termitomyces heimii. So we have got 3-4 species here that we recognize, and different above ground or epigeal stages of Termitomyces. Now, how do we understand the various characteristics of the epigeal or above ground stages of various species of Termitomyces? In the previous few video clips, I have given details of the morphological oddities like this mushroom, so I will not repeat that. Let me first focus on the egg stages or the immature stages and give certain comments regarding the very interesting aspect of the melanization and the melanized epicuties or the melanized hormonal region. Whichever egg stage we take, we can see now this dark part, which is nothing but the fungal melanin. Now, it is very much essential for the epigeal stage to protect the very sensitive developing reproductive tissue, that is this tissue. This needs protection, and this protection is afforded by the universal veil. So the universal veil is surrounding this entire, means it is a sort of a natural seal, and the natural seal is protected by the melanized tissue. So the tissue produces melanin. We don't know much about the localization of melanin, whether it is cell wall bound, or it is in cytoplasmic, that is not known, but our reference to known literature clearly shows that there are melanin granules which are likely to be found in hyphal cell walls, which make these epicuties. So we see that because of the pressure, this was originally a perforatorium, so all of these were in the pseudorhizal stage. You see here that this is a fruit body which is developing, but this has just drilled through very hard soil and it has become black because the melanization has made it tough. So the epigeal stage, if we detect more and more dark color, blacker and blacker hormonal region, it indicates the transition through a tougher layer of soil. And also this type of feature will be possible only when the perforatorium breaks through a soil of uniform toughness, so that you can see even the tip here, it is very black and it has got lacerated, completely cut off and peeled off. So everywhere we see that there are marks left due to the struggle which the growing fruit body at young stages has to drill through. So it is not known whether the fruit body can rotate like this and become distorted, because what generally we assume is when this is transiting from the basal attachment, reaches pseudorhizal stage, then forms a stipe, then it will just go in a linear fashion, that it will go like this and then will come up. But we do not know whether any twisting or rotation like this takes place while it drills. Although it is believed that something like this may happen, otherwise it is very difficult to actually explain this type of laceration, these type of remnants of the universal veil which we find in the epigeal stages of the cap. So these lacerations are possible only if the fruit body rotates like this while drilling the young fruit body, whether it is drilling and then while drilling it emerges. So if it is drilling, then it would be very interesting phenomena in mycology, because then we will get an example of the presence of certain forces. Like here we see that we have got a very conical type of hormonal region, and again this type of pattern. Now this pattern is not possible unless this fruit body is rotated slightly like this. So whether the driller acts like some sort of the mechanical drill, that is not known, but from the intensity of melanization of these fruit bodies in the young stages almost, and also the attachment of the soil here. We can see the force that is exerted by the driller because the soil is tightly bound to this, and from this we can make out that it is not only wherever we get this attachment binding of the soil here, we can see, for example, this is a distorted stipe and a distorted cap. Now obviously when it was trying to come out, it got oriented because there was some sort of obstacle in the path of the developing pseudorhiza, so it got slanted and entire stipe has become eccentric. So actually this is a very interesting oddity because the stipe in genus Termitomyces should be central. So here we give an example of a typical central stipe, and here we have an example of an eccentric stipe which is attached on the side. Now why this deformity occurs in apigeal stages? That is because of the type of the mound soil or the overlying material through which the driller has to drill through. So the apigeal stage is fully dependent on the transient driller stage. And we see the example here also, there is a distortion here because of the toughness, and you see the umbo is like a tip of the rocket, and it has become black and it has become very thick and leathery and tough. Now this toughness gives its mechanical integrity and the required force because it has transited, it has travelled so much from here to here, and then further we see that a fibrous stripe, and the stripe has also got this sort of the damage here because it is not due to insects but the fight, the struggle which the fruit body has to do, the apigeal stage has to do. The growth towards light, growth towards wind, and perhaps the rains means hydrotropism, positive hydrotropism, negative gravitropism, positive thigmotropism, and most important positive phototropism, because all these species are definitely phototropic. Although we don't know much about the photosensitive, the mechanism which is possibly present in genus Termitomyces, where the photosensitive sensors lie, we do not know. Where environmental sensors lie, we have no idea whether they lie in ambonal region. This part is acting like a biosensor which senses change in humidity, which senses the direction of and the intensity of light or the sunlight flux, whether it senses humidity, whether it senses temperature and acts like a temperature sensor, we do not know. But without such information, it is difficult for the fruit body to emerge, and then if it emerges, you see the eccentric stripe is there in another specimen also. Whereas in specimens which we may consider as healthy, normal and not deviant, because this is a deviant specimen, and this is a normal specimen with a normal typical morphology, textbook I would say specimens, textbook apigeal stages, absolutely, textbook gills, pale pinkish. So these are textbook wild type specimens. Where the distortion occurs? The distortion occurs in the apigeal stages because of several environmental and other features. So this is a distorted stripe, this is an eccentric stripe, this is a lacerated stripe, this is a stripe which is mechanically has suffered a lot, and you can see the insect damage and all that, and still it has struggled to come out. So apigeal stage also becomes an extremely important aspect of Termitomyces fruiting. In this entire life cycle, not only it is the hypogeal stage which we have already described in our previous video clips which we have recorded, this is the hypogeal stage, but apigeal stages also have to struggle a lot to come up and grow to this stage and then successfully develop the reproductive organs in these gills and then complete their intended genetically programmed life cycle. So we have normal specimens like this which are just like textbook, which have pseudorhiza, stipe, centrally this one central attachment, pink gills and smooth and ornamented type of cap, and we have these poor, distorted, deformed apigeal stages. another deformed. So now if you keep these deformed stages in one group here, if you keep these deformed stages in one group, can we say that these deformed stages qualify for description under separate species? Definitely not, because we know that there are sure reasons which we can look at the habitat and explain that these have encountered some very tough the soils or obstacles, so that's how the stripes got distorted, and this can be explained by making detailed field records. So what we suggest is, in terms of understanding Termitomyces ecology or biology anywhere in the world where scientists, ecologists, mycologists, research scholars may be going, our advice is, do not jump to the conclusion that just because this is an odd specimen, you see how it is convoluted, it is spirally oriented, and then this is like a very beautiful that folded a pygmy like a specimen. Do not jump to the conclusion that you have come across a new species of Termitomyces. Try to record the local characteristics which has made it go round like this, maybe because of absence of light, maybe because of the change in rainfall pattern, maybe because of overlying vegetation, maybe because of some branches of trees or something touching the cap, and that's how it got distorted. Maybe it was a piece of rock around which the stipe had to go around and then give rise to this fruit body. So all these obstacles will have to be taken into account to explain the distortion of the apigeal stipe stage. Before we conclude that no, this is a different species, no, we cannot jump to the conclusion that it is different species. It is a different way the species have tried to adapt itself to overcome the obstacle. So when we look at the collection of these interesting specimens from 3 different species, Irisus, then Medius or Gumantakiensis, or Entomoloides, which are smaller species here, which are some of the, and this also is an oddity because you can see the width of the stipe, and these features we have already recorded earlier from the campus. Because of certain environmental consideration, these oddities occur. So this throws a spanner into the whole idea of species plasticity. That means if environmental conditions change, the morphology also changes. So that becomes a very interesting challenge for Termitomyces taxonomists. Here our focus is to lay down certain emphasis on the importance of melanized region of the apigeal stages. It is the melanin which protects the species from all types of attacks, microbial attacks, against various types of hard obstacles, against radiation also, and that makes the apigeal stage of Termitomyces feasible and possible. So in the next this one, we will see some more interesting aspects of Termitomyces ecology and biology.